All right, everyone. Welcome back to our last session. Before I do not introduce Nick, because we don't have introductions, I just wanted to, I don't know that anyone said anything at the end of the last session about how fantastic the student presentations were. That session is always one of the highlights, and this year was no exception. And so I just wanted to applaud all of them. I didn't have a chance to get the microphone back to say how great that session was. So they set a very high bar before our last talk of the meeting, and after which we'll all go for drinks and then the social dinner tonight. So without further ado, I will turn things over to Nick.
I'm sweating because those were such great talks, and my fellow speakers did such a great job. So thank you so much for inviting me. It's a real pleasure to be here. I've never been to Rovereto before, to this meeting. I've really enjoyed it. I think I have the benefit of going after all of the fabulous talks that we've heard, so I'm going to take some liberties with some aspects of this work that you've heard from other people. So the topic of what I want to focus on today is prediction. And prediction in a sense that might be a bit stretching the term from what people typically mean. So most work on predictive coding is on what I would call perceptual prediction. This is where your expectations, oh yeah, 2 microphones, not good. This is where your expectations are about the future or current identity of a stimulus, an individual stimulus. So to give you a sense of some instances in which these kinds of expectations manifest, I thought I'd show a couple of demos. So one is this classic illusion. This is an apparent motion demo. Who sees these dots as moving left to right, horizontally? Put up your hand if you see it moving horizontally. Okay, who sees it moving vertically? Okay, so what if I do, you see it horizontally now, okay. But what about now? Right, so stimulus is the same, but something internally is guiding you to think of it. Okay, I can do it again. Focus on it. Weak minds. Okay, you got it locked in? You looking? All right. Oh! Mel got it, thank you. If anybody's stuck, all right. All right, it's just Sharon. All right. This is, if anyone's locked horizontally, now hopefully this will help you see it vertically. Maybe. Oh, good. So the point is the stimulus is the same. You're bringing something to bear on your interpretation of the stimulus. This has also been studied in cases of amodal completion. You should hopefully, modulo Sharon, see this as a kind of light blue disk superimposed on these inducers, even though there's no color in the middle here. It's the same white on the outside as it is on the inside. You're filling in essentially something that's not there. There's also something called modal completion. So in this picture, you probably see this woman kind of in front of the fence, her dress in front of the fence, and this man's jacket in front of the fence. The color and brightness and so on of the dress and of the fence are the same. So the fact that you see this as a contiguous dress and not as a contiguous fence is an assumption you're bringing to bear, in particular that you tend to fill in edges that are short. So the edge cutting off the fence here is shorter than the edge that would be cutting off her dress, right? So these are cases where you're bringing to bear some assumptions about the world to infer what you're seeing about an ambiguous stimulus or about a stimulus where there's missing information. The mechanisms of how this kind of expectation work in the visual system thought to be top-down feedback through a hierarchy, say in visual cortex, that has some higher-level interpretation that's then projected down to constrain lower-level processing. So in the case of this kind of classic Kanitsa-type illusion, you have these individual inducers that you could process as individual objects, but some higher-level recognition of this triangle shape induced by the inducers projects down and actually leads you to perceive this as a triangle, including to represent the edges that are missing on the boundaries between the occluders. This kind of filling in has been studied in the visual system. This is a paper from my postdoc, whose work I'm going to talk about later, that he did in graduate school, where he basically shows that V1 represents these illusory contours. When you use a reconstruction model from fMRI data, you can see that there's activity at the location of the contour. So this is kind of the standard way that people think about predictive coding as feedback within a cortical hierarchy that leads to higher level interpretation that then constrains lower level interpretation. What I'm going to talk about is a slightly different, kind of looser sense of prediction, what you might call mnemonic prediction or memory-based prediction. And this is where expectations of a stimulus result from learning of an arbitrary relation or a set of arbitrary relations with other stimuli. That is, where the properties of the current stimulus don't necessarily tell you what's going to show up next in their features, but rather based on prior learning that's occurred. One example is what happens when you crack open an egg. There's not much on the outside of an egg that tells you what's going to look like on the inside with white and a yolk. Another example is when you walk in your front door, or as you're opening your front door, you have expectations about what furniture, what room you might be in, but there's nothing about the look of this door that tells you what the furniture will be like inside. You can think of navigation this way. When you turn a corner in a familiar navigational route, you might know what buildings to expect or what people you might be likely to see. Or inspired by yesterday's presentations on voices, if you see somebody's face like Sharon, you have some sense of what her voice is going to sound like, even though what her face looks like does not tell you exactly what her voice sounds like. This is a relatively arbitrary mapping. So the question is, how do these kinds of expectations arise, and do they have the similar kinds of consequences for visual processing as these sorts of more perceptual effects that I was showing before? The framework that I want to introduce for thinking about this, what you might call sort of a representational hierarchy. This is a familiar figure to most of you, the Fellman-Van Essen diagram of sort of wiring in a primate visual system. This is old now. It's been updated in many ways, but this is still the old version. One thing that often goes ignored is that the top box here, HC, is the hippocampus. So that's kind of the endpoint of the ventral visual stream, and in some sense the dorsal stream. And the idea is that whereas standard predictive coding really focuses on sort of how higher level interpretations or mid-level interpretations of a stimulus constrain lower level visual processing, I want to make essentially the same kind of argument that when you get up into the temporal lobe and the hippocampus, the nature of the representation allows you to form these memory-based expectations that then project back down and influence visual processing. So whereas as we saw earlier in Nicole's beautiful talk, whereas inferior temporal cortex might represent the identity of an object or the category of an object, when you get into the medial temporal lobe, what governs the representations in the medial temporal lobe is not the visual features of a stimulus, but rather their spatiotemporal features, whether two objects co-occur together in time. So whereas two books or two lamps you see at different locations at different points in time might have a similar representation in inferior temporal cortex, that's not true in the hippocampus. Rather, a lamp and a book next to it might come to be represented more similarly because you tend to see those together. So this system is able to learn arbitrary relations in a way that received and feedback to influence lower level visual processing. So that's just the general framework that I'm working from. I think the hippocampus can serve this role among other systems. I mean, people talk about medial prefrontal cortex and other areas as well, but I'm going to focus on the hippocampus for three reasons. The first reason is that the hippocampus receives inputs from all over the brain. It's a convergence zone. This is the site of the hippocampus, if you don't know it, and then this is sort of the circuitry of how the hippocampus interacts with a subset of its possible projection partners. So in red are the subcortical connections from the hippocampus. In black are the cortical connections. This is a vision-biased wiring diagram, so you see T and V4 projecting in. But in fact, all sensory modalities project into the hippocampus. This is how, for example, you're able to form really rich episodic memories. When you smell something familiar and it evokes a sort of rich memory of your mother making your favorite dish, or when you hear somebody's voice, knowing what their face should look like. The hippocampus gets all of these kinds of projections from different systems. So it has convergent inputs from different kinds of information. So as we were talking about before, parietal might represent object information, parahippocampal might represent spatial information, hippocampus can form sort of object location bound association, even though those are relatively arbitrary inputs. Given that it receives these convergent inputs, the next sort of critical property of the hippocampus, from my view, is that it's able to rapidly integrate these inputs into integrated or merged representations. I'm going to show a study from my lab here just to illustrate this principle. And so the idea is that when inputs to the hippocampus co-occur regularly, they get combined together into a joint representation. So this is the work of former graduate student Anna Shapiro. And Anna did several great experiments on hippocampal learning, particularly statistical learning in the hippocampus. But one of the more basic ones, just to illustrate this principle, is where we showed people a sequence of fractal patterns that look like this. Their task was to detect every once in a while one of the fractals would have a grayscale patch on it. So it was a cover task. What we didn't tell them, while we were showing them these for 45 minutes, what we didn't tell them is that the order with which the fractals appeared was structured. So after the first fractal, you were 100% likely to see the second fractal in a pair. Those were embedded in a continuous sequence. So there are no segmentation cues. The only way that you could learn these pairs is by picking up on the transition probabilities in the sequence. We also embedded, sort of as a control condition, what we called weak pairs, where the second item in the pair followed the first a third of the time. And the hypothesis here, along the lines of what I was hinting at below, is that the mere fact of pairing two fractals in time repeatedly will cause the hippocampus to represent them as more similar to one another. The co-occurrence in time is a cue that they may be part of the same event or episode. The way we tested this is by presenting that sequence of fractals, but before and afterwards taking a snapshot of the representation of each of the fractals. You heard about this technique in the presentation before mine. So we presented all the fractals several times in a random order, before and after exposure to these pairs, measured the pattern of activity evoked in different regions of interest for each of these fractals, and then looked at the similarity of the patterns of activity evoked by fractals that were paired strongly and weakly. Under the assumption that exposure to these regularities has shaped the representations to combine them into some sort of integrated memory, the pattern of activity evoked by the two members of the strong pair might be more similar than to two members of a weak pair or two fractals that were just recombined randomly. We looked at this with high-resolution imaging. This was one of our first high-resolution studies, so this is, the methods now are even more sophisticated, but this was essentially high-resolution anatomical imaging, where we're able to manually segment different subregions of the medial temporal lobe. This is sort of the hierarchy from visual cortex into parirhinal, parahippocampal, entorhinal, which provides the input, you see here, to the hippocampus. The hippocampus is built out of several subfields, including the dentate gyrus, the CA3 subfield, the CA1 subfield, and the subiculum. We can trace these subfields based on known anatomical landmarks at very high resolution, and then essentially extract the population of voxels from each of these subfields and look at whether exposure to regularities has changed the similarity of the patterns of activity evoked by the fractals. Just because I'm just hoping to illustrate this principle, I'm just going to show you the final sort of most compressed result. This is the change in pattern similarity evoked by fractals, now appearing in a random order, but that had been paired before during exposure. The change refers to an increase in pattern similarity from before to after exposure. So up means integration, down means differentiation. And I'm going to plot three kinds of pairs. The shuffled pairs are just a baseline where you take two fractals that didn't have any special relationship and you combine them, sort of a permutation test. You see no change in the similarity of activity patterns. However, for the strong pairs and across all three of the subfields that I'm showing here, we saw increased pattern similarity for fractals that had appeared together reliably in time. I'm not going to go into it today, but the weak pairs actually turned out to be quite interesting. These shuffled pairs had a baseline probability of about 2%, so they were very unlikely to pair together. We kind of figured the weak pairs would be somewhere between the strong. That is, things that appeared together 33% of the time might be sort of part way. In fact, what we saw was reduced similarity of the representations evoked by fractals that had been paired together only a third of the time. So this is a non-monotonic change in the representational similarity as a function of probability strength. And we have ideas about this I'd be happy to talk about if people are curious. The point is that after less than an hour of exposure, the representational structure of the hippocampus came to mirror the statistics of the sequence that it had been exposed to. Okay, so I'm just showing one study. Anna did a lot of other work to really characterize the nature of this learning, including looking at more complex naturalistic kinds of structure, so where the sequence is drawn from some underlying graph that has interesting properties. We also did some patient work in collaboration with Mike and others, looking at a patient named Lonnie Sue Johnson who has bilateral hippocampal loss and who is impaired at statistical learning, in fact doesn't show any statistical learning. And finally, just last year, Anna published a beautiful theoretical paper using a biologically plausible model of the hippocampus to look at the kinds of representational change that I just illustrated in a simulation where we can separate the dynamics of the subfields and look at how two sort of primary pathways in the hippocampus encode episodic memories and statistical regularities. So the point of this is just this is that second sort of piece of evidence the hippocampus might be playing this role of getting divergent inputs, combining them into integrated representations. And then the final piece of the puzzle is that once you've learned these kinds of representations, they can then be used in the service of prediction because of a property of the hippocampus known as completion. So memories can be retrieved, these memories can be retrieved based on partial cues. The effect of this is not just to retrieve the memory in the hippocampus and to make you aware maybe that you've seen something before, but also to reinstate the retrieved content back into cortex. This is thought to be what the experience of remembering is returning sensory systems to the same state they were in as during encoding. So what it means to retrieve a memory is to send some information back into the systems that encoded that information in the first place. There are several studies on this that I'm not going to go into, but that have looked at, for example, the extent to which hippocampal activity at retrieval drives cortical reinstatement of features that were present during encoding, and then how that influences your ability to report that you've seen something before. I also put in a study from Alexa Tampari, who's over here, who did some really beautiful work on reinstatement and MTL cortex. For example, looking at the extent to which, when you retrieve something, if the pattern of activity evoked during retrieval mirrors what was present during encoding, you might be more likely to remember that information. So these encoding retrieval similarity analyses have been used to support the idea that the hippocampus drives cortical reinstatement of encoding-related activity. So this is all work essentially from the memory literature that I think can be used as a way to think about how the hippocampus could support representation of expected information in the visual system. I'm going to talk about two studies that are more directly focused on prediction. The first has to do with cross-modal expectations. So if you hear this, you kind of like start looking around and thinking about where's the ambulance, is it coming at me? If you're driving, you've probably all had that moment of looking in your mirror to see if you're getting pulled over. The sound of an ambulance in this case retrieves some sort of percept of what kind of vehicle you might see. In the second part of the talk, I'm going to talk about another kind of mnemonic prediction, which is how actions shape our expectations about what we should see in the environment. I gave the example before about cracking an egg or opening a front door. Those are cases in which your actions in the world determine the sensory outcomes of what you experience. And so to what extent do actions shape hippocampal representations and hippocampal prediction? An example that I like to use here, given that we're close to the mountains, is you can imagine being at the top of a ski hill, getting a cue like this, knowing with my poor skills that I should turn right or turn left and suffer. And then depending on what action I make, that might influence my expectations of, say, like the steepness of the slope that I'm going to encounter. So given some cue in the world, I can decide how to act. Action will disambiguate what I should see next. So that's the kind of learning that we'll talk about in the second part. Okay, the first study was work of Peter Cook, who I mentioned before from his graduate work. He was a postdoc in my lab for the last couple of years. He's starting his own lab at UCL this summer. And Peter did a lot of really beautiful work on predictive coding in his dissertation. I thought I would show one study just as a setup for the study that I'm going to talk about today. So this is a study where he essentially trained associations between tones and oriented gratings. So he trained people that 75% of the time you heard a low tone, you would see a rightward tilted grating, and if you heard a high tone, you would see a leftward tilted. The actual task is that you would hear a tone, then you'd be presented with two gratings that were very similar to each other, and you had to do a same different judgment. Okay? But essentially, hearing a low tone allows you to set up an expectation that you should see something tilted to the right. What he was looking at this study is what do these kinds of expectations, these cross for visual processing. People before had shown this too, that when you expect a stimulus, in this case the green, when you can expect a stimulus, that leads to overall reduction in bold activity in V1 in this case. This is called prediction suppression or predictive suppression. But there's two ways of explaining this. The standard sort of theory of predictive coding, what you might call explain away, is that when you have an expectation, that serves to or inhibit your the expected stimulus. So that if what you're expecting actually shows up, it doesn't drive a net increase in activity. If you know what's going to happen, there's no need representing that information from the world. Instead, by suppressing the expected stimulus, you become more sensitive to errors, opportunities to learn what you weren't expecting. In that case, explaining away says that you have reduced activity because you're sort of blocking the stimulus from activating its representation. There's a different account, what you might call a sharpening account, which is almost the opposite and is equally prevalent in the predictive coding literature, which says that when you have an expectation, that allows you to filter out noise. If I want to listen to some radio station, I tune the dial to that frequency, and that has the effect of letting me hear the signal at that frequency and not noise from other frequencies. So expectation, actually, from a signal processing standpoint, allows you to hone in to filter the information in a way that you're more sensitive to the information you're expecting. In that case, you might also predict a reduction in activity because you're suppressing noise, right? So both of these accounts were used to explain these sorts of prediction suppression effects. Interestingly, though, they make opposite multivariate predictions. In the explaining away case, you're suppressing the expected orientation, so you should have less orientation information when the grading you get is expected versus unexpected. In the sharpening case, you should have enhanced information about expected stimuli compared to unexpected stimuli that have been inhibited as noise. And so he applied the kinds of decoding techniques that you heard about today and showed that despite an overall reduction in bold activity, there's actually increased information about expected stimuli in V1. More consistent with this sort of sharpening idea that expectations serve to enhance stimulus information rather than inhibit it. So I mention this just because this is what I think of as a very beautiful result, showing that expectations, even cross-modal expectations, influence very low levels of sensory. Maybe why I came to my lab is great, but where are these expectations coming from? Visual cortex is not encoding arbitrary audio-visual associations that you've trained for 5 minutes, right? There has to be some sort of joint coding of this arbitrary relationship. Note that these aren't well-learned multisensory associations either, right? This is an arbitrary thing. In fact, halfway through the experiment, he reverses the contingencies to show that it's not a stimulus effect. So the very simple hypothesis, which should be predictable given the setup that I gave you, is that given a situation like this where you've learned that a low tone is associated with this grading, when you hear that low tone, that might bring to mind a representation, an integrated representation of tone and grading in the hippocampus, which then in visual cortex. So that's the hypothesis that we wanted to test, is to what extent does the hippocampus retrieve associated visual information from auditory cues, and how does that relate to expectation effects in visual cortex? We didn't use a decoding approach. We tried to use an approach that's a little more generative than that. In fact, we used what's called an inverted encoding model. And the idea is that when you are looking at a continuous stimulus space rather than decoding left versus right, you can take a pattern of activity, say in the hippocampus, and infer what it's representing, not just classify its activity. So just to explain this a little bit better, we created a novel shape space using a technique called Fourier descriptors, so if you have to explain if anybody has questions. And we associated one point in the shape space with a pattern of rising tones, and a different point in the shape with a pattern of falling tones. So whenever you heard rising tones, you would see this shape 75% of the time. When you heard falling tones, you'd see this 25% of the time. The other 25% of the time, you'd see the other shape. Okay? Now, with this sort of technique, we basically ran the same kind of task that I introduced before. So on the critical runs where we're going to look for hippocampus, it was the same sort of thing as what I had before, where you hear a rising scale, then you see two shapes that are either identical to each other or subtly different. The way that they differ, orthogonal dimension to the shape space that's defining the predictions. So when they were different, it was a very small deviation. This is just asked to make sure that people are attending to and potentially benefiting from the expectation. Okay, now to train up the model that'll let us kind of do reconstruction of what a hippocampal pattern of activity means in the shape space, we had additional localizer runs where we presented each of the shapes on their own with equal probability. By doing that, we can measure the response of every voxel to each of these shapes. So just to show it here, when you sort of scan people and are showing them all these different shapes and you measure the average pattern of fMRI activity in response to each shape, you can then learn via regression a set of weights of how well, in this case, 5 hypothetical channels or tuning curves explain the activity of a voxel across those shapes. So you essentially learn a set of weights for each voxel of how much of its activity is explained by each of these 5 tuning curves. With that sort of, that's the encoding model part of it. We've learned a tuning curve for every voxel about how it responds in the shape space. Then given some test item, now in the prediction runs, so where we show, say, we measure the pattern of activity over voxels, say in the hippocampus or anywhere else. We can then take the weights that we learned from the encoding model, take a pattern of activity over voxels, and infer the amount of evidence for each of those channels or tuning curves in that test pattern. This lets you reconstruct in shape space what information is present in a pattern of bold activity. So it's like a decoding or MVPA kind of analysis. Instead of classification, you're actually generating the stimulus rather than decoding the stimulus. So how well does this work? Well, what I'm showing you here is V1, just to prove that we can reconstruct shape information despite these relatively subtle differences in shapes. So with a test pattern, here's the sum over the channels in response to a shape presented at this point in shape space. So what you see is there's a lot of evidence for onto the shape that was presented. So this is basically showing that we can reconstruct from a pattern of activity which shape they're looking at. Not so surprising. If we had the other shape that people could see, we can reconstruct that shape very well as well. And we can quantify something like how well we're reconstructing this shape as the difference in the evidence, say, when you're presented with this shape versus this shape at the correct shape versus at the incorrect shape. So the difference in evidence here tells us how much kind of item-specific information we're reconstructing from the bold activity pattern. So we have some measure now, reconstruction, applied to the hippocampus. If you remember, 75% of trials were valid trials where what you were expecting actually showed up, and 25% were invalid trials. Now, there's two things that the hippocampus could be representing. It could be representing what your expectation is, or it could be representing what stimulus you're shown. On valid trials, you can't decouple those two things. So if you were given the rising tones that predicted this shape, that's both on the valid trials, the shape that was cued by the tones, and the shape that was presented. So you can ask a question, though, even in this case, is it even possible from patterns of hippocampal activity, where it's hard to find stimulus-selective responses, to reconstruct the identity of both predicted and shape? And in fact, that we can reconstruct stimulus-specific information from the hippocampus. Now, the more interesting question is what happens on the invalid trials. Lets us dissociate the predicted shape or the expected shape from the presented shape. So now you might be cued with the decreasing tones, but actually get the shape. This is just doing sort of sensory processing. It should represent the presented shape and look kind of like this. If it's representing the expectation, it might be centered on the cued shape, even when that's not the shape that's presented. And in fact, that's what we find. The information in the hippocampus is dominated by the cue, the presented shape. Just to show you how strong a reversal this is, it's a full crossover. It's really totally dominated by the expectation. This differs from V1. If you do the same analysis in V1, which you might expect to be more stimulus-driven, in fact, you find that it is. I've collapsed it in the same way now, and so it's centered on the same shape. And so regardless of what you're expecting, V1 represents what you see. There is some modulation of what you see when it's inconsistent with what you're expecting. That's what this difference in the tail corresponds to. So there's an effective expectation on visual cortex, but it doesn't dominate the representation. Just to show that these effects are selective, we ran the reconstruction model and searchlights over the whole brain. Information about the presented shape is in visual cortex. Information about the cued shape is in the hippocampus. And there's a smaller cluster in the caudate as well, which I'd be happy to talk about. OK, we can do a little bit better in this and do the kind of hippocampal segmentation that I told you about before. I didn't go into the details of the circuitry, but we had a hypothesis that these effects might be stronger in the CA3 subfield of the hippocampus. This is the subfield that does the kind of pattern completion that I mentioned before, that takes a partial cue and retrieves associated content. Partially consistent with our hypothesis, we did see reliable decoding of the expectation within the CA2-3 dentate gyrus subfield. This is combined because people don't think you can separate them, but reviewers don't think you can separate them. So it's combined. But we do see evidence for reconstruction of the expectation in that subfield. We also funnily saw it in the subiculum, which is sort of the output relay from the hippocampus. So maybe what's being retrieved by CA3 is then being sent through the subiculum. OK, so now what about the interaction between those expectation signals and sensory processing or visual processing? We did find, as I hinted at before, modulation of visual responses by expectation. Here's LO, for example. This is decoding that difference in the reconstruction curves for validly versus invalidly cued shapes. And what you see is that the fact that green is higher than red here means that there's information about the presented shape earlier when it was validly cued by the tones compared to when it was invalidly cued. In fact, on the invalid trials, negative here means that you're representing the other shape, the predicted shape, rather than the perceived shape. So you see the cue sort of drives the representations in the wrong direction, and then you recover. So the fact that you see this difference here suggests that the cues are facilitating, or at least maybe in the case of the invalid, delaying information about the presented stimulus. The question is, how does this kind of facilitation in V1 or LO representations in this case relate to hippocampal retrieval of the associated shape, that kind of prediction signal that I showed you before? What I'm plotting here is an across-subjects correlation over the whole hippocampus between the shift in the latency of the shape signal in LO and the strength of about the cued shape in the hippocampus. And what you see is that there's a reliable relationship there. If you then further break that down into subfields, you see that this correlation is dominated by the subfield that we hypothesized would be important, and not so much by the subiculum, which showed a main effect but no correlation. So this is tentative evidence, correlational evidence, that something about what the hippocampus is doing related to retrieving the associated shape has some relationship with the extent to which your expectation influences processing of the shape in LO. Now, building on that, we can do the same sorts of analyses, but now incorporate into the equation. This is work by a former postdoc, Nick Hindi, who now has his own lab at University of Louisville, actually Sharon's former graduate student as well. The idea here was to control for stimulus transitions, but make actions the. So just to illustrate the structure, on predictable trials, you would see QA. If you made a left, you could make one of two responses. Either you could press the left button or the right button. If you made a left response, 95% of the time you would get stimulus B. 5% of the time you get stimulus C. If you made a right response, 90% of the time you'd get C, 5% of the time you get B. Okay? So if you ignored what action people are making, B and C are equally likely. A goes to B half the time, A goes to C half the time. But if you know what action they're making, you can predict with almost perfect certainty what's going to appear next. You can compare this kind of condition to a condition where the actions really don't have any impact on what appears. So in this case, when you press left, you're equally likely to get both fractals. In this case, when you press right, you're equally likely to get both fractals. Again, in terms of the stimulus transitions, the predictable and unpredictable condition, when you take actions into account, you can almost perfectly predict the outcome in the predictable case. You're going to have to take my word on it. I'm not actually going to show you any of the unpredictable data, other than to say that everything I'm about to show you does not work in the unpredictable condition. And if you subtracted out the unpredictable condition, you would get the exact same results as I'm showing. Okay, so here's the hypothesis. Let's go back to the sort of pattern completion hypothesis. Imagine you see that AQ that you've learned has some relationship to two outcomes depending on your action. If you see the AQ and you make a left action, our hypothesis was that the hippocampus would retrieve this integrated memory based on this partial Q of seeing A, pressing left, and getting B. You would have a sequence memory in the hippocampus, and that this pattern completion in the hippocampus would allow you to represent the expected outcome in visual cortex. So you can see a similar kind of logic to the previous study. Here we can do something which helps isolate the expectation a little better. We have three kinds of trials. On one kind of trials, it's similar to what they did during training, where they see A, they press left, they get B most of the time, or they see A, they press right, and they get C. We had other trials where we just showed them the outcome, and they didn't make an action, either outcome B or outcome C. And the critical trial type here was like that intuition I gave you a moment ago, where you see the Q and you make an action, but then nothing happens. So these Q plus action trials really let you isolate the expectation because there's an omission of the expected outcome. The approach we're going to take here is a slightly nuanced classification approach. We're going to use those other two trial types, the full sequence trial type and the outcome trial type, to train classifiers that are sensitive to sequence memory in one case and to outcome information in the other case. So just to show you that there are expectation effects in this study, let's start with the outcome piece of this. We train a classifier based on the trials where you only see an outcome. So we train a classifier to distinguish B versus C. Then we're going to test it on the trials where we show the Q, have people make the action, but then the outcome doesn't appear. To the extent that they're generating an expectation of the outcome, we should be able to classify these trials into the proper B and C category, right? If you see A and you press left, if that reinstates B in visual cortex, we should see evidence for B in the classifier, and vice versa. What I'm showing you here, again, is sort of a simplified version of the results from the critical subregions. These are the same as before, so CA1, CA2, 3 dentate gyrus, and subiculum. And in visual cortex, we use FreeSurfer to segment V1 and V2. This is the output of the classifier at test. So it's trained entirely on outcome trials and is tested entirely on Q plus action trials. Evidence for a prediction here would be that we can classify these test trials accurately in visual cortex. And in fact, we find that. These aren't whopping effects, but remember, this is item-specific decoding of a prediction. This is about as specific a kind of decoding as you could do. We didn't necessarily expect outcome information on its own in visual cortex because we thought the hippocampus was retrieving the sort of full sequence of activities, so where you saw A, left, B, as opposed to just B on its own. And on just B may not be sensitive to that full sequence information. And in fact, we could not decode the outcome on its own from the hippocampus. Okay, so now let's focus on the hippocampus part of this, sequence decoding. Same logic, except now we're going to train a classifier on the full sequence trials, A left B versus A right C. Train the classifier up, and then again test it on the very same Q plus action trials. To the extent that this partial Q, the Q plus action, retrieves a sequence memory, we should be able to classify these partial Qs into these full sequences. Again, applying the same classifier in both visual cortex and the hippocampus, we can now reconstruct from CA2-3 dentate gyrus and CA1 the sequence to which the Q and action correspond, but we cannot accurately classify the full sequence from visual cortex. So just to be clear here, these are the same test trials in both cases. What this means is the hippocampus is representing sequence information, visual cortex, or at least these visual areas, are representing outcome information. What's the relationship? I showed some correlational data in the previous study. Do the same kind of correlational analyses here. We were able, because we had more power in this study, to do within subject analyses as well. So that's the first thing I'm going to show you here is across trials within subject, according to our sort of general hypothesis, the extent to which the hippocampus retrieves the correct sequence should determine whether the correct outcome appears in visual cortex. Or at least that would be consistent with that kind of causal mechanism. And indeed, that's what we find. There's only information about the outcome in visual cortex on trials where the hippocampus retrieves the correct sequence. We can also do a cross-subject correlation. Are people who show stronger sequence memory in the hippocampus, are those the people who also show more outcome information in the cortex? And indeed, we find a strong relationship there. I generally avoid trying to make temporal conclusions about from fMRI data because of the sluggishness of the bullet response, but we were prompted to do so by a reviewer, and so I'm going to show you the data because I think they're consistent with the hippocampus driving visual cortex. In particular, these trials lasted for a few seconds, so we could train a classifier over time within trial. And our prediction, according to this sort of hippocampus as a source of expectations, is that retrieval of the sequence in the hippocampus might precede in time, or should precede in time, the appearance of the outcome in visual cortex. Whether we can see this in fMRI is a separate question, but mechanistically, that's what we think is going on. Compared to the reverse, that's equally plausible with the kinds of correlational data I showed you, that somehow representing the outcome in visual cortex drives sequence memory in the hippocampus. So you can look at, we can sort of regress and try to predict, in this case, the outcome information from the sequence information over time, from one volume to the next, or the reverse, try to predict the sequence information from the outcome information. And we see that you're only able to reliably predict in this direction, which is consistent at least with this sort of reinstatement. We're currently doing intracranial studies where we have electrodes in the hippocampus and cortical surface electrodes in visual cortex, where we're going to be able to get much higher temporal resolution and hopefully solve some of these temporal precedence questions more convincingly. We're also ramping up to do stimulation in the hippocampus, which could selectively knock out the kind of pattern completion cortical reinstatement, and we would predict that that would knock out expectation information in visual cortex. Okay, so I think I'll skip. I was going to show one more quick finding, but I'll skip ahead for the sake of time and summarize. The hippocampus, this has been widely known for many, many studies. The hippocampus forms relational memories, in this case of multimodal sequences, including stimuli and some action information. This allows missing or upcoming stimuli to be cortices based on partial cues. This link between pattern completion and visual expectation is consistent with the role of top-down statement, as has been argued for low and mid levels of visual processing, maybe now for higher levels of visual processing. And so in conclusion, from these studies, pattern completion and cortical reinstatement seem to be a potential putative mechanism for prediction based on learned sensory relationships. Just to end on a sort of maybe a more higher level point, how these kinds of studies have influenced our thinking. The hippocampus, as many of you learned about it, is a system that does declarative, implicit, episodic memory. And I think this kind of work, and many, many other studies in the last few years, have suggested that maybe that's not, that kind of mapping of a construct onto a brain region might not be the right framework for thinking about what it does. Rather, we tend to think of the hippocampus as supporting a set of computations that are maybe unique to this system, and that different tasks can load on those computations in different kinds of ways. So for example, I've talked about relational binding. The ability to learn from a single experience associations is very unique. This idea that the hippocampus is a convergence zone. Pattern separation, which I didn't talk a lot about, but which is really essential to hippocampal function. This is the idea that spatiotemporal properties govern representations in the hippocampus. So if you feed the hippocampus two similar inputs that occur in different contexts, even if their visual features are nearly identical, it'll orthogonalize those inputs and store them as discrete memories. That ability to take overlapping inputs and orthogonalize the representation is very unique, and is, I think, the reason that hippocampal representations can be governed by spatiotemporal co-occurrence rather than feature overlap. We talked about pattern completion and its role in retrieval, cortical reinstatement, the fact that hippocampus outputs to visual cortex. And finally, there's even a theoretical idea, at least, that the hippocampal circuit itself might be doing predictive coding and prediction error detection. Specifically, CA3 projects retrieved information to CA1, but CA1 also gets sensory information from visual cortex. So CA1 is well positioned to a differencing sort of operation where it compares expected information to experienced information. There's actually not great evidence for this, at least in humans, but it's an interesting idea. And more generally, this is how I think the field of memory is going, and has allowed people to think about the hippocampus in a more integrated way as being part of the ventral stream and maybe part of many other systems and circuits. So there have been nice review studies that say instead of sort of labeling brain systems based on tasks or cognitive constructs, we should instead think of sort of computations that they support. And we've written about this as well, how the hippocampus is used in many different kinds of cognitive tasks. So I'll stop there, and thank you very much.
Great talk. Thank you very much. It's really interesting work. A lot of what you're talking about as a function for the hippocampus here reminds me a lot of what's been spoken about for basal ganglia function. Yeah. Caudate tail in particular, right? These are structures that are really fundamentally involved in this sort of sequence learning. Do you see any evidence for a relationship? I mean, one of the extreme there would be that the hippocampus is actually showing you something that's being created by these other subcortical areas. Yeah. And have you had a chance to look at that, or the involvement of caudate in these sorts of tasks?
Yeah, so we often get caudate activity, univariate activity, these sorts of learning tasks. There was sort of an older idea that the hippocampus and basal ganglia, striatum, other prediction task where observational learning was hippocampal, but trial and error learning was striatal. I think that story has also changed a lot, and they're operative than people previously thought. Wouldn't be surprised if some interaction, more striatal effects in our studies and other motor sequence component or motor learning component, and when the timing between stimuli is fast. Sort of variable timing, longer delays. Even kind of hard to caudate. Great point. And it's definitely, I would be surprised if it wasn't part of a circuit that can do kinds of things. Yeah. Yeah, well, so there's two. One is the time scale of learning. In the case of the striatum has a longer time scale of learning, so a lower learning rate, a slower learning rate. And in addition, there's the timing of the stimuli themselves. So there seems to be an optimal rate for basal ganglia processing, maybe like 600 milliseconds, whereas the hippocampus in trace conditioning studies and other studies can bridge over long variable gaps. So in both of those senses, the learning rate and the stimulus presentation rate, I think you might get interesting associations between the systems.
So yeah, I want to ask a related question. So gorgeous work, and continuing on that notion. So now we have this does, and I'm interested in hearing your thoughts on what the hippocampus doesn't do. Yeah. So you've talked about cross-modal type associations. That's an obvious case. But going back to the case of vision, you've also talked about visual associations that are arbitrary. Yeah. So is there a non-arbitrary association, visual association, that is somehow reflected and that is not hippocampal mediated, or is it all hippocampal mediated? And so, you know, as a toy example, how about motion extrapolation, right? That just the prediction that this thing is going to keep going, and so I'm going to predict it's going to end up there some future point in time.
I sort of view this mechanism as being on the same continuum as the standard sort of predictive coding mechanism. So if the prediction can be generated locally within visual cortex, I think that the hippocampus may not be relevant. So motion extrapolation is a great one. The Kanitsa kinds of illusions, I would be surprised if those were hippocampally dependent. Peter would kill me for saying this because we're just looking at the data now, but he basically ran his grading prediction task as opposed to shape prediction task, and hippocampus seems to be differently involved in the case where the expectation is about a feature rather than an object. It's too early to really sort of definitive what that difference is, but that seems to be relevant at the complexity or the level at which the expected information needs to be represented, the degree to which the campus is involved. Yeah. Also, how well learned the association is. So I would imagine that you could consolidate these kinds of associations, and then they could become hippocampus dependent. Yeah.
Hi. You showed the Peter Cox study with the gratings, and you showed, you nicely presented this distinction between sharpening and suppression. And you find this improvement of decoding in V1 for expected gratings, and you seem to not find that in your second study where you use shapes. Why is that?
So do you mean the improved classification of, I see what you're referring to now.
You had one graph where you had V1. You also, the same in LO actually, but.
Yeah, this is like kind of a supplementary figure that gets to this. So this is the reconstruction for the shape study in V1, V2, and LO. The difference in what he found is he found greater peak information about expected stimuli. We find earlier information about expected stimuli. So there is this difference. That is a difference between the studies. He found greater overall information. We don't see a higher peak decoding signal, but it does come on earlier.
Do you think it has something to do with the fact that there are shapes versus gratings?
It could be. That's why we ran the study at Nicole, was to do rising tones, the whole hippocampal thing, with the acquisition parameters we used in the shape study, but with a grading prediction to see if we would get the same kind of time shift or an increase in peak information. Jury's still out on that, but I think the time shift is very reliable. It showed up in all of these different parts of visual cortex.
And in your action, in your action study, it seems like you had these left versus right actions that were decodable in hippocampus, but how can you distinguish that from the actual motor action?
Yeah, good. Well, that's where that non-predictable condition comes in. So they're still making left versus right actions, but you can't decode the sequences when the actions are not predictive. So yeah, it's a great question.
I guess what I wanted to ask a slightly more general question about the function of hippocampus versus other brain areas. You mentioned at the end the important role of the hippocampus in pattern separation, where what that essentially involves, I guess, is taking items that may be similar at one level of representation and separating them at other levels of representation. It seems to me that that's something that has to be done in lots of different forms of cognition, sort of resorting similarities, right? Things that are similar visually may not be similar conceptually, or things that are dissimilar visually may be similar conceptually. Perhaps easiest to see when you're doing language processing, where you're translating, auditory inputs, two words that may sound very similar but mean things very different, or vice versa, or the same thing visually. So, and of course a lot of thoughts about how that processing goes is that you're transforming from one level of representation to another, in part because you need to shift the representation such that the similarities reflect the properties that you need, not the surface properties, but the deeper properties. So I guess the question is this something, would you argue the hippocampus is involved in that, or is this notion of recomputing or moving things around in vector space is just a basic function of a lot of neural computation?
It's a great question. It is a basic function of cortical computation. The idea that you sort of squeeze and stretch representational spaces comes up in perceptual learning and language. We saw earlier category learning. What the hippocampus does that's unique is it can do that from a shot. if you have an experience today and then you have a similar experience tomorrow, that experience tomorrow will be assigned a unique representation. The kinds of learning that drive representational change in cortex that induce cortical differentiation or integration generally slower and more gradual. And so you don't get immediate orthogonalization. It might take several rounds of prediction error. It might take feedback of different sorts. So there's a lot of evidence for, say, acquired equivalence. If two things predict the same thing, they come to be represented more similarly over time. If two things predict different things, they come to be represented more differently. That's typically slower and more gradual. So I think that's one key difference. And the extent of the separation is really remarkably high in the hippocampus from a single attempt. To get that kind of large-scale orthogonalization in cortical representations, I think would require a lot of training. But yeah, I think it's a general purpose idea that the representational space in lots of different brain systems have to come to conform to the content in semantics, in language, in vision, in task space. Yeah.
I don't understand what you mean by reinstatement.
Yeah.
I have an image projected on a part of the retina. It was one part of V1. Mm-hmm. And now you reinstate. What do you reinstate and where? Now, did you probe to see if spatial information is fundamental? In other words, I don't understand what it means to say, if I show you a square that is this size, then I show you a square that is only this size. Or I show you a square here versus here, or orientation here versus here. Where is this stuff reencoded, and what is being reinstated? Do you have any ideas about that?
Yeah, I mean, a lot of people have worked on this, but I could give you my sense of this. The idea, at least the theoretical idea in memory, is that you are recapitulating the representations that were active at encoding. And so if you encoded a memory where there were two parts in space and you present one part of space as a retrieval cue, you would see information show up at the other part of space. Mechanistically, neurophysiologically, whether that's the exact same representation or not is not known, and I doubt that it is the exact same representation. But the idea that there's some sort of internal drive to create missing information is what people sort of think of as episodic retrieval reinstatement. I showed one example at the very beginning where you got filling in along edges in V1 that were missing in a Knizia illusion. So that's sort of an operational sense of it. I don't know if that helps.
No.
So tell me what you need.
Well, I'm not a vision scientist, so I'm trying to reason about this from principle. Yeah. So if it is the case that when I see a grating over here, yeah, I'm activating neurons that have those properties here. Yes. When I reinstate it, and you show me now an image not here, but over here, okay, how does it help me?
Where you mean you show the same location
yes.
now? So yeah. There's no information missing in that sense, right? You show something and then you're showing it again. The sense of reinstatement is where there's a partial cue. So imagine you have a full screen scene that I show you, and then I show you the left side of the scene. The idea of reinstatement is that there would be some sort of filling in of the right side of the scene. The idea, I agree with you, if you take a stimulus and you move it over here, showing that there's information that goes back there, I don't think that would impact your ability to perceive that stimulus in the second attempt. It's really about creating missing information or generating information that you're likely to encounter next.
So let me ask my question differently then. If you did have that condition, then you would predict it shouldn't get an effect.
If you had which condition?
In other words, if I show you the stimulus here,
yeah.
And then when I test you, I put it over here, there should be no benefit in V1.
You would not get the same priming if you moved it to a new location. There are other reasons why testing would be different. I just don't think the hippocampus would have anything to do with that.
The claim is not that it's always in V1. No. I think he's not claiming that reinstatement.
We were talking about V1 and V2. I was just referring to the data about V1, V2. I wasn't talking about the other areas, just V1, V2. I was curious about what the claim is.
Is like the you know, it's like the, let's see if I can pull it up quickly here. You know, this is literally the idea that like, you learn this sequence a lot, then at test I present you with a cue and I have you make an action. That allows your hippocampus to retrieve the sequence memory. This fractal, I haven't shown it to you, it's blank right now, but now this fractal is active in the hippocampus, and the hypothesis that we have some evidence for is that there's information about this fractal back in visual cortex. That's the literal operational meaning. I don't know.
Right, so what I find fascinating about this is that it claims, or it seems to claim, that in the hippocampus, you have a veridical representation of that thing.
Yeah.
That is, you have to assume that the representation in the hippocampus is absolutely as clear as it is in V1, in some sense.
Yeah,
so
that is an assumption.
That's a very strong assumption.
But
and so what is the evidence for that?
That's the assumption of episodic memory, though. When you have a very rich recollective experience, you know, I don't know, a very salient event in your life or a memorable meal, you have at least the feeling that you are experiencing, you're able to reminisce about things that aren't present, and they have vivid details. Some of that is elusive, you know, illusion, but we do have a veridical feeling about prior experiences.
I think it's important to clarify this, and then I'll stop. Yes. That's good for him. It's good for his heart. So let me finish.
This is a very memorable experience.
This is very good. Right. So just one question. Exactly. So I think that the interesting issue then is if one were to play with, say, slight distortions or whatever of these kinds of representations. You see, I always thought that perhaps people believed, and I am not a neuroscientist, people believe that representations in the hippocampus are not that specific. Okay, so if they're not that specific, then there must be some mechanism for generating the specificity to show effects in V1.
Yeah,
and so I guess
I think this is interesting. I think they are that specific. Which is why we have rich recollective experiences. It's also the case because of that pattern separation function that we were talking about before. If you inverted these colors or you made it a different size, it may not actually trigger retrieval of the same memory. So this sort of question about generalization is an interesting one. Yeah.
I just wondered when you decode information in V1, whether it's in world or in retinotopic coordinates. So if you shift fixation during from training to test, do you find now you can put it in the other hemisphere, or is it somehow?
It's an interesting question, I think. So
sort of related to the question that Alfonso raised, really, about the nature of the representation that's being reinstated in V1.
My guess is it's in world coordinates. I don't know of a study that's looked at that, but it's typically about content. If location is somehow an important feature of the experience, you know, and its retinotopic location was somehow important.
Yeah, so if you've always been fixating here and presenting stuff here, and now you move the fixation point to the other side, then things should, I guess, shift over in visual cortex.
Yeah, I think if your expectation is spatially specific.
Yeah.
Yeah, it's a good
well,
question.
world specific, not spatially specific. It's out there in that particular part of the world, yes, but not that particular part of retinotopic space.
Yeah, I think part of the reason why it wouldn't be retinotopically specific is that the learning in the hippocampus may not be retinotopically specific object representations.
Yeah.
Although there's some evidence for retinotopic coding and for temporal cortex from Julie Gollum's work and other people. So yeah,
right. It would be most useful to be in world coordinates.
Right, I think it would be.
Yeah.
So I'm wondering to what extent the predictive coding in the hippocampus depends on actually experienced temporal contingencies, or whether it can be influenced by other kinds of knowledge. So for example, maybe I see Sharon and I know what voice to expect, but if I then see her inhale from a helium balloon, then I'll have a different expectation that I didn't actually experience before, but that's kind of a modification of the expectation based on other factors. Do you think that hippocampus predicts those kind of influenced predictions as well, or only actually experienced ones?
I would say that this is one of several systems that have the property of being able to activate representations that aren't currently present. So for example, a lot of work on prediction is semantic prediction, semantic expectations in a novel experience, right? I walk into somebody's kitchen, I have some sense of what the objects are, what I can do with them, what's going to happen. That can be represented as an event script, that could be represented as a schema. Moshe Barra's work on sort of low spatial frequency representations of expectations represented in anterior temporal lobe, semantic representations, along with Mike saying before, part of the purpose of shaping, crafting cortical representational space is to get activation of related, information or concepts. So I think that there's many factors that can feed back and influence processing. The hippocampal contribution, in my mind, is the ability to arbitrary relationships, not pre-existing cortical knowledge structures, rapidly. So we're talking in many of these cases minutes rather than days or years. And it gets these projections. So cortical learning of this sort requires representational overlap. You can't get purely arbitrary associative learning across domains in cortex unless the areas are multisensory to begin with, right? So you could get cortical binding of disparate inputs if there's some co-location to begin with, whereas the hippocampus really can take everything and do it in a very general way. But yeah, it's a cool question.
Sorry. So to get back to sort of the sequence learning that you were discussing early on, reviewing Anna Shapiro's work.
Yeah.
So there was that interesting little data point you mentioned you might be able to get back to later for the weekly associated items, which was like, 1 in 3 times they would be followed by. So what is the explanation for that? Is it that the hippocampus is coding an expectation that it probably won't be that item?
I think that's a great way to, we're done. No, that's a great way to state the intuition behind it is there's some non-monotonicity, no relationship to strong relationship, and then the thing in the middle is below baseline, right? So you have to have kind of a 2-factor explanation in that account, which is there's some minimal amount of relationship that you try to learn about it, and you discover that most of the time it's not what happens next. I think I just blacked it out. There we go. So yes, I think that's a conceptual way of describing what's happening. Mechanistically, what we think is happening, we were talking about this at lunch, is there are learning rules that are non-monotonic that can explain these kinds of effects. Ken Norman has been doing a lot of work in this space. We have a grant together to look at these kinds of effects. This is exactly what Mike was asking about before. That effect is a differentiation effect. So seeing things together a third of the time makes them less similar. Seeing them together 100% of the time makes them more similar. Why is 33% less similar? In these kinds of non-monotonic learning rules, partially activating a memory makes it susceptible to weakening. This is true in reconsolidation, retrieval-induced forgetting, many other sorts of tasks. So in the case where you have a weak association, you may be sort of predicting, and that weak prediction, especially in the case where other things show up most of the time to compete with it, can have the effect of degrading that item. I didn't mention, but we also inserted the 2/3 of the time that the outcome didn't show up. In that case, we presented them randomly in the stream at other times, which allows the representation of that item to sort of adopt features of the new context in which it appears. So I think it's that sort of moderate activation, weak prediction that's driving differentiation from the first item in the pair and allowing it to shift in representational space away. That's a quick version of it.
Okay, thanks. Yeah, I was also interested in that that study you did with Anna. When you so the strong transitional probabilities, was it always A goes to B? It was never the other way around? Okay, and so then what changed pre to post?
I wish I hadn't taken that slide out, but I'm going to do something.
So
Sharon asked me this question like 3 years ago, so I have a slide on it.
Thank you, Sharon.
You did. Do you remember that?
Yeah, I mean, what?
Yeah, it's a great.
Presentations become more similar post, but what's changing?
Yeah, so in the CA3 subregion, thanks for coming, Melissa. Your baby was very well behaved. In the CA3 subregion, because of this temporal order, we had the sort of same idea that the first item might reactivate the second more than the second reactivates the first, because what happens after the second could be anything else. What I showed you is that these are just more correlated with each other, but how are they changing? So one way to look at that is to say, how do they start off? And essentially to see, does the first item become more like the second, and the second becomes like the first? Is there some sort of temporal asymmetry in the movement in representational space? And you do see that in this sort of CA3 subregion. Okay. It's weak and noisy. You also see it in the subiculum, but it's in that direction. The first adopts properties of the second more than the reverse.
Thank you. That answers the question.
All right. Is this on? Yeah? Okay. I'm going to ask the last question because I've been sitting here very quietly for 25. What? I thought we'd go quarter till. Is it quarter till? Yeah. I have quarter till. Yes. Okay. Okay. What? Okay. But I mean, this could be a 5-minute question, the rate I'm going. I have a question that I think might be a variant of Alfonso's generalization question, but Mel was distracting me by waving at me over and over. So I apologize if I'm repeating it. But it has to do with, again, my general confusion about where what the hippocampus does stops and where what cortex does starts, or the other way around. And it's partly motivated by how you analyze your data, which is looking for one particular pattern at the expense of all other patterns. So, take, for example, my prediction now that it's 5:45, so I'm likely in a few minutes to see an Aperol Spritz in my hand. But it would be equally, maybe not equally likely, but could happen that there's going to be a glass of white wine in my hand, right? So it might be that my hippocampus is going to just be predicting the most common thing to what's going to be in my hand at 5:50. And so it would actually it would show a big difference, huge difference between, and this might be your pattern separation point, between my very well-defined spritz patterns and wine patterns. But in cortex, the important thing is to know the category of things that might be coming. And this goes back to your very opening examples of what you're likely to see when you go in the door, right, of your house, or the ambulance that's coming. Yep. So it seems that your methods aren't well tuned for finding predictions of categories or of schemas. They're really well tuned for finding predictions of instances, and it could be that's why your data are all pointing at the hippocampus. Yep. But if the real problem of prediction that we face as learners is predicting categories of things, you're missing out potentially on what the rest of the brain is doing.
Yeah, I totally get what you're saying. So semantic information is, most regularities we know that are useful for prediction are of the form you're talking about. No experience is ever the exact same. It's a different time, right? And so we need to generalize in some way. The examples, the framework is the role of essentially episodic memory in guiding expectation. I think that's part of the equation. We can have very specific instance memories of the last time I was in some place this happened. And that's sort of a framing issue. I don't think that mechanistically that's all that's going on. The campus is some instance, as you describe it, but it gets fed back into a system that's not organized based on instances. So you can get generalization in the feedback through, say, inferior temporal cortex or anterior temporal lobe or wherever. It would spread, or at least transfer to related information. So we haven't tested it. I think you could test this empirically. You could look at reconstruction of out-of-sample members of the same category based on episodic learning. So one prediction would be if you see A and you retrieve B, if B is related to CDE, is there information about CDE there, even though they've never seen them before, based on your prior associations between B and C and D? Yeah, yeah. So that would be really interesting.
No, he just answered.
Yeah. This is so beautiful that we should have discussion. You said that the things become more similar. In what sense? I mean, what does more similar mean in this context?
Okay. That's not a quick question. The question is quick. The answer is not quick. There are two things you could learn. We've looked at this. It's very hard to get at this with fMRI. Really, what it comes down to, if you're asking what's learned in the hippocampus, there's two kinds of things you could learn. right? You could have take the pair, right? You could have one representation of A and a representation of B, and what the hippocampus could learn is some new representation that contains information about A and B. That's how we think about episodic memory. You have some cortical object representations, and you're combining them into some sort of configural or relational representation. In that case, you're retaining the individual representations of A and B. They're linked to a combined representation. So, an example for seeing the first member of a pair will retrieve this memory of A and B together, which could then prime retrieval of the B item, right? That's one way you could learn. That's what happens in I apologize for sighing, it's just there is a longer version of this talk. Anna did some really beautiful modeling work exactly looking at these questions of how representations change. I'll just show you the summary of her model just to be able to talk about it a little bit. That's what happens in this circuit. You retain discrete representations of each of the objects in CA3, and they get additionally linked to a configural representation. This green pathway here, it's called a monosynaptic pathway. The one's the trisynaptic pathway. The monosynaptic pathway people hadn't looked at before. It actually merges the pre-existing representations of A and B. So the difference is, as a result of training, here you have A, B, and A plus B. Here you have like the average of A and B only. And that's based on the learning rate and the nature of the representations. It's a really interesting question, those different kinds of the implications of those different kinds of representations. So behaviorally, for example, A and B become more confusable after this kind of learning. So if you present a rapid scheme that has a bunch of A's and B's in it, and you say, how many A's did you see? In this kind of learning, you overestimate the number of A's. In the case of the weak transition, part of the time, you actually underestimate the number of A's. So that's more consistent with the sort of merging account. How far that goes, I'm not sure. But yeah.
All right. I think we'll wrap up there. Thank you, Nick.
Thank you.
On behalf of all of the members of the organizing committee, I want to thank all of the speakers, the attendees for great questions, and for the poster presenters, for the award winners for their great talks. So just a big round of applause for everyone who shared their science with us over these 2 days. Yeah. I also, on behalf of Mel, Alfonso, and Alex, I want to give a special shout out to Marius. We're all listed as if we're equal contributors to pulling this meeting off on the poster, but we all know that that's just not true. So normally we embarrass Marius at dinner tonight by thanking him, but he's not able to make it to the banquet tonight. But you all should make it to the banquet tonight. Be back here in 2 hours, and thanks again, Marius, for all your work on this. Anything else to say? All right. Go get your drink of choice, fire up your hippocampus, and be back here in 2 hours.